I'm Dr. Vijay Reddy. I'm the Chief Medical Officer for D2V Clinical. I'm a licensed physician in the United States, and I have worked in the biopharmaceutical industry for many years. My career in antibody drug conjugates, or ADCs, began many years ago while doing my Ph.D., when I developed monoclonal antibodies that were conjugated with the chemotherapeutic drug doxorubicin that targeted CEA-expressing colorectal cancer cells. Advances in ADC design and technology have rapidly increased the pace of approvals in recent years, with over a dozen ADCs that have been approved, with many more on the way. ADCs are designed to combine the benefits of targeted therapy and chemotherapy. The three components of ADCs, to remember broadly, are the vehicle, which is the monoclonal antibodies that delivers what's called a payload or chemotherapeutic drug to the cancer cells that have a target, which is a tumor-associated antigen. Early phase clinical development lays the foundation for the success of ADCs, and careful evaluation has to be given to the target identification, patient selection, safety, and dose optimization for efficacy. The best example to understand how critical the early phase development can be understood by the development of the first ADC that was approved, gemtuzumab oxazomicin, or Mylotarg for acute myeloid leukemia. While it was originally approved nearly 20 years ago, it was subsequently withdrawn voluntarily due to safety issues. However, after further dose optimization with a lower dose and different scheduling, it was found that the benefit outweighed the risk, and the FDA approved it 17 years after its initial accelerated approval. Therefore, early phase clinical development is critical in understanding the target. As examples, certain targets such as CD30 are ubiquitously expressed by cancer cells in the case of Hodgkin's lymphoma here with brentuximab vedotin. In the case of HER2, ADCs targeting HER2, there are 15 to 20% of breast cancer cells express this, and can also be, however, effective in a so-called bystander effect, where HER2's null cells which have no HER2 expression, can benefit. Therefore, understanding the payload and the target are critically important. Recent ASCO conference demonstrated promising results for several ADCs that are being evaluated in clinical trials. Examples of these ADCs include targets such as B7H3 in metastatic castration-resistant prostate cancer, HER3 in metastatic breast cancer, TROP2, HER3, CCAMS, MET, etc., and non-small cell lung cancer. In conclusion, ADCs are antibody drug conjugates that combine the benefits of antibodies and targeted therapy with chemotherapy or cytotoxic agents. Early phase ADC clinical development is a dynamic process and is driven by dedicated professionals like you. If you're working with an ADC development program and require expert guidance, we're here to help. Contact us today to schedule a consultation and talk to our experts regarding the ADC development program. Thank you.